Bismillah ar-Rahman ar-Rahim. Welcome back to the content video of Genetic Material chapter represented by me, Dr. Farah Fadwa bin BELGACEM from UD-BKM. By the end of this chapter, you will be able to recognize the history behind DNA discovery, describe different structure and states, distinguish between DNA and RNA, and relate genetic material to cell life. DNA, this amazing creation responsible for passing traits from generation to next, is composed of two strands of nucleotides forming a spring-like structure called a double helix. However, this double helix is not perfect. It all began in 1869 when the Swiss scientist Frederick Miescher discovered an important substance with phosphorus content is called nuclein. Then the Russian biochemist Febus-Lever came 1919 with the discovery of nucleotide, which is composed of phosphate group, a sugar, and a nitrogen-containing base. The Austrian biochemist Erwin Chargaff added to science the famous rule where the amount of adenine is equal to thymine, and same goes to guanine and cytosine. By the early 1950s, the American biologist James Watson and the English physicist Francis Crick discovered the 3D structure of DNA of double helix using the X-ray photograph of DNA taken by the physicist Rosalind Franklin, who unfortunately passed away before 1958 and was therefore eligible for Nobel Prize given to Watson and Crick. For a better understanding of the genetic material, we must first understand the chemical nature of DNA, which consists of subunits called nucleotide, containing a deoxyribose sugar and a nitrogen-containing base, which both of them form the nucleoside. When attached to a mono-D or triphosphate group, they make the nucleotide. Chemically talking, the bases adenine and guanine belong to the purine chemical class of two-ring structure, while cytosine and thymine are members of pyrimidine with single-ring structure. When comparing between DNA and RNA, the first obvious difference is the double strand characterizes the DNA, while the RNA is a single-strand form. The sugar is the second difference as the DNA has a deoxyribose but the RNA has a ribose. Last but not least, bases are similar except the thymine present in DNA and uracil present in RNA. To know about the structure of the nucleic acid, the most important part is how the nucleotides are linked to each other and how the double strands of DNA maintain the double helix shape. The adjacent nucleotides in the same strand are covalently linked by what we call phosphodiester bond, while the two strands of nucleotides are held together by hydrogen bonds, in which adenine are opposite to thymine and cytosine are opposite to guanine. The organization of DNA in chromosomes, the cell genetic content is called genome. It consists of one or more chromosomes. Each chromosome is a single double-stranded molecule of DNA. Humans, for example, have 46 chromosomes per cell. In the genome, we distinguish two states of the genetic material, chromatin and chromosomes. The chromatin is the material that makes up a chromosome. It consists of DNA and protein called histones, which play the role of packaging and condensing the DNA molecule, while the chromosome is one complete object, which may contain up to thousands of genes. During DNA replication, each of the two strands that make up the double helix serves as a template from which a new strand is copied. The new strands will be complementary to the parental or old strand. When the two daughters' DNA copies are formed, they have the same sequence or are divided equally into the daughter cells. The process of DNA replication employs a large number of structural proteins and enzymes, each plays a role or critical role during the process. The replication starts from what we call the origin of replication, which is a sequence of around 245 bases, very rich in AT bases. An enzyme called helicase unwinds the DNA by breaking hydrogen bonds. As the DNA opens up, a Y-shaped structure is formed. The topoisomerase, an enzyme which is present to avoid DNA to wind or to supercoil. RNA primases then synthesize an RNA segment to let the DNA polymerase primes DNA synthesis. Then the DNA polymerase extends a new strand of DNA. This happens only in the 5' to 3' direction. Since the other strand has opposite direction, small fragments of Okazaki are formed away from the replication fork. Here the ligase enzyme is needed to attach the Okazaki fragments. After the chromosome has been completely replicated, the two DNA copies will be moved into two different cells during cell division. That's all what we need to know for the moment about the genetic material. Keep watching our videos for the next chapters. Thank you for your attention.